Hello, students.
Good afternoon, sir.
Afternoon, sir.
Good afternoon. Good afternoon. Okay, in the previous class, we started that biomolecule and the questions related to that. Okay, so today we will continue that one and some questions. Please see that. Did you do this question that which is given number one? In a protein structure, the first amino acid and the last amino acids are respectively called as this. Did we do that? In the protein chain, okay? In the protein chain that which is made of amino acids, the one say if in the chain there is so many amino acids are there, and the first one, what is the name of the first one and what is the name of the last one? This is N-terminal. See, options are N-terminal amino acid and C-terminal amino acids, or C-terminal amino acid, N-terminal amino acid, alpha amino acid, beta amino acid, beta amino acid. Which one? Hello?
Number
Number? The correct option is, see, when the amino acids are joined, say different amino acids are joined, then what happened? During this, the amino acid are joined by which bond? They are
peptide.
joined bond by peptide bond. Is it? And in the peptide bond, what happened? That the carboxyl group of one amino acid and NH2, this is amino group of the next amino acid, is it? Amino group of next amino acid, they join together and form the peptide bond. So what happened in the amino acid? So this is H, this is R, this one is N, this side is C, OH carboxylic group, and this side is
Wow.
NH2 amino group. So if this one is the first amino acid and the next amino acid, they will join. These two will take part in the bond formation. Is it? So first amino acid having always this amino group is free for the first amino acid. So this one is the first amino acid is known as N-terminal amino acid. First one is N-terminal, and having the second amino acid, that is, if we see, this second amino acid is this one always will be free with carboxylic group. carboxylic group. So this one is C terminal. And in this way that it continues. They say this is first one, this is second one, the third one will join. The next peptide bond will form between the carboxylic group of the second amino acid and amino group of third amino acid. So again, the carboxylic group on the right side, it will be free. So it is the C terminal. So our answer will be that the first amino acid is known as N-terminal amino acid and last amino acid is C-terminal amino acid. A polypeptide is always have at one end it is N and the other end it is C. So C-terminal and N-terminal. Okay. So this is the answer. Next one. So here the acidic amino acids carry two carboxylic group and one amino group per molecule. Keeping this in mind, select the correct pair of amino acid. Correct pair of amino acids. Say the character of all the amino acids, they depend on three things. Okay, what are those three things? Say this carboxylic group, because this is acid, carboxylic acid, no? So this gives acidic nature. This one is amino group is basic. So carboxylic group and the amino group, and the third one is R group. This one is fixed, this one is fixed, this is fixed, and there is changes in this one. Okay. So here, if this R group is acidic, then the amino acid will be acidic, because this one here with these two, this is acidic in nature, this is basic in nature, so they balance it, neutralize it. Okay. But here, if it is acidic group, the amino acid will be acidic. If here it is basic, then it will be basic. If this one is neutral, then the amino acid also will be neutral. So amino acid may be acidic, amino acid may be basic, or it is neutral, based on how many amino group or carboxylic groups are there in the amino acids. So this is here in this one, aspartic acid and glutamic acid. These are the correct pair, that they are the acidic. All right. This is acidic. They are, so there are acidic and basic amino acids. We should remember the different amino acids. Okay, which one is basic, which one is acidic. Okay, I will show you some portion that, see here, this is based on the number of amino and carboxylic groups. Okay, based on the number of amino and carboxylic groups, so there are acidic, basic, and neutral amino acids. So acidic amino acids are aspartic acid, glutamic acid. All right, this is acidic. Basic, lysine, arginine, histidine. Neutral, alanine, lysine. Sorry, there is lysine is neutral. Valine, phenylalanine. These are the neutral amino acid. This is not correct. Okay, this is not correct. So, and aromatic, they have aromatic, this one, tryptophan, phenylalanine, and tyrosine. These are the aromatic amino acids you have to remember. And then some amino acid, they have sulfur in them, and sulfur containing amino acids are cysteine and methionine. Okay. Basic, lysine is basic. No, lysine is, this is correct. This is correct. Here it is lysine written is wrong. This one is wrong. Okay. This one is wrong. Lysine is basic. Here, so you have to remember this chart from the book. Then you can remember that which is acidic, which is basic. Next question is elimination of water moiety during the formation of peptide bond. You always remember that there is a water molecule. There is water is removed during the formation of this bond. That is the say this biomolecules, they are usually they are the polymer. Okay, whether it is protein, this polysaccharides, polysaccharides, nucleic acids, they are polymer, and they are formed by joining the monomers. And during this formation, during joining the monomer, different types of bonds are formed. But during the formation of this bond, always one molecule of water is released. Say in the peptide bond formation, one molecule of water is released. During the formation of the glycosidic bond, glycosidic bond is formed where? Glycosidic bond is formed in the formation of polysaccharides. Okay, when two monosaccharides are joined, there is a formation of glycosidic bond, and during that time also one molecule of water is released. In case of nucleic acid, there is formation of ester bond. Okay, ester bond, and during this formation of ester bond also there is release of water molecule. So remember this during this polymerization that water is released, and this is the dehydration, not the hydration, this is dehydration. So our answer is dehydration. Here is one question given that which one out of A to D, this is A, B, C, D, given below correctly present the structural formula of a basic amino acid. In the basic amino acid, what will be there? In the basic amino acid, one carboxylic group and more than one amino group. Is it? So, which one? Whether this one is, this is carboxylic group 1, amino group 1. Is there any other? This is carboxylic group. Okay. COOH. This one will be acidic. This one acidic group, one basic group. So it is neutral. This is not the amino acid. This is not representing amino acid. Okay. This one carboxylic group, one is amino group, and see here another amino group at the end. In the R chain, this is the R group. Okay, in the R group there is another amino group is there. So amino group means it is basic. So our answer will be option one is C. C is not the answer. Option 2 is D. So our answer is 2. Next, I think this one perhaps we did in the previous class. Quaternary structure referred to the spatial relationship between, in the quaternary structure, the polypeptide chain in a multi-chained protein. Quaternary structure is more than one polypeptide. More than one polypeptides are there. So this one, which is incorrect. Quaternary structure referred to the spatial relationship between individual polypeptide chain in a multi-chain protein. This is correct. Tertiary structure is absolutely necessary for many biological activities of the protein. Okay, this is true because tertiary structure that achieve the 3D organization. Biologists describe the protein structure in 3 levels. No, this one is wrong. So biologists describe protein structure in 4 levels. Okay, these 4 levels are primary, secondary, tertiary, and quaternary. Okay, so there are 4 levels of things. So this one is incorrect. So here is our protein structure is correlated with protein function. Naturally, every molecules or everything, so their structure is suitable for their function. The function it has to do or the function it carry out, that will decide their structure. So this is all true. Here is one: match column 1 with column 2 and choose the correct option. Acidic and basic. Say acidic is here glutamic acid. Basic lysine. Aromatic are these: tyrosine, phenylalanine, tryptophan. These are the aromatic. And neutral is valid. So what will be 1 with 3? 1 with 3 is this one only. B, B2, yes. C is 1 and D 4. So this one. This 2 is A2, A4, A1. No, this one is the correct. Next, essential amino acid. One thing we should know about, one term is there, that is essential and non-essential cell amino acid. Essential and non-essential amino acid. So here is two questions we have here related to the essential and non-essential amino acid. So one is essential amino acids are those which are not synthesized by our body, not required by our body, synthesized by our body, and only used to form protein. Which one?
Option 1.
Number one, sir.
Number one, not synthesized in our body. These are the amino acid which the animal body mainly they cannot synthesize. Okay, so we have to take it from outside. Outside means we have to take it from through our diet, through our food. Okay, but it is essential for our body and not synthesized, and we have to take through dietary protein, through our diet we have to take. Similar way, here it is given another question. The lysine is an essential amino acid. Okay, lysine is an essential amino acid. Why it is essential amino acid? It is rare? No. It has high nutritive value? No. It is important constituent of all protein? That's also not correct. It is not formed in the body and has to be provided through diet. Okay. So here we have a list of this So here, this is one essential and non-essential amino acid. This is the list. So these are, here it is arginine, histidine, but leave these two: isoleucine, leucine, methionine, phenylalanine, threonine, tryptophan, lysine, and valine. These are the essential amino acid. Okay, and these are non-essential, but here non-essential does not mean that it is not required for our body. These are also very, very important important amino acid for us, but these things our body can synthesize. Okay, and this our body cannot synthesize. These two amino acids, this arginine and histidine, actually during childhood it is required, but in the adult this can be synthesized. So they are kept in a semi essential type of uh, amino acids. Okay. So you have to remember the list, which one. Now come to one. This is a question from the carbohydrate. Okay, saccharides. Here two diagrams, diagrammatic representation of two molecule of sugar given, and you have to identify what are these. This is given, this is A, this is B. So here is A, ribose and glucose. Is it ribose and glucose or deoxyribose, ribose, glucose, ribose, ribose, deoxyribose?
Option C, sir.
Option C,
C.
glucose and ribose. This is the five carbon sugar. This is pentose, is it? This is pentose sugar, and this one is hexose sugar, is it? They have carbon 1, 2, 3, 4, 5, and this is the sixth number. And here it is 1, 2, 3, 4, and 5. This pentose. This one is, this is the ribose, and this one is glucose. Okay, glucose has a open chain and closed chain structure. This one is closed chain structure, ring structure of glucose. Okay, this is a ring structure of glucose. Pentose, these are always a ring structure, and this in this case, the in the pentose, this first carbon, first one, third one, and this fifth, these are very important. In case of this is ribose sugar. Okay, in case of deoxyribose, in case of deoxyribose, this oxygen will be, missing, absent. That's why it is also known as not only deoxyribose, 2 deoxyribose. means 2-deoxyribose, oxygen from the second carbon is missing. Okay, so if you just change this one, it will do. So here is answer this one. Next one is given here, again the structure of the molecules, and you can see this is one structure, this is one structure, this one is one structure. This one is same. This one is only sugar and a nitrogenous base, adenine is attached. Sugar and nitrogenous base. This is nitrogenous base, and this one contain sugar, nitrogenous base, and phosphoric acid. So what is this? The first one, this is a nitrogenous base. Nitrogenous base, and when it is nitrogenous base plus say here the nitrogenous base, next structure is the nitrogenous base plus sugar, this form
Nucleosides.
nucleosides. And here that nitrogenous base plus sugar plus phosphoric acid.
Nucleosides, sir.
It is a nucleotides. So this one is nitrogenous base. This nitrogenous base is what?
Adenine, sir.
Adenine. Adenine, adenosine, and adenylic acid. Adenine, adenosine, and adenylic acid. Adenylic acid is the adenine, adenosine, adenylic acid. This one. Say this is adenine. Adenine has do you know this one, this position is important. This is number 1, 2, 3, 4, 5, and 6. This is this sixth position is very, very important. This sixth position, adenine and cytosine. Remember AC. Okay, they have amino group A. So you can remember with A is amino. At the sixth position, they have amino group. And guanine, thymine, and uracil. This they have they have a keto group. If you remember that AC, that they have amino group at the sixth position, the Rest are with the keto group at the sixth position. Okay, here in this position, there will be keto group, like it will be like this way in place of this amino group. And this adenine and guanine, of course, adenine and guanine, so they are purine, so they have a side chain also. And the pyrimidine, they don't have side chain. This is the pyrimidine. Okay, this one is purine. This is pyrimidine. So here also you have to identify A and B. This is amino group at the sixth position. So it is adenine. This one is adenine. Okay. And this one is what?
Uracil, sir.
Yes, it is uracil. This is uracil. So you have to decide between this thymine and uracil. Okay, which one is thymine, which one is uracil? Actually, uracil is the base structure, and thymine is 5-methyl uracil. 5-methyl uracil means in this position, this is 1, 2, 3, 4, and this is the fifth position, this is 6. This fifth position, in this uracil, it is simply, it contains 1 hydrogen. But this one will contain, in case of thymine, it is 5-methyl uracil. At the fifth position, there will be a methyl group. Right? If this is replaced by a methyl group, then this one will be thymine. Again, cytosine will have this one NH2 amino group. Okay, so here it is adenine and uracil. Oh, this question has been disturbed, so I'll leave this one. Due to I'll leave it. All have been okay. But here is one question. This one, the selected portion. Okay, you can say select the correct option which represent the homopolysaccharide made up of glucose. The homopolysaccharides made of glucose is this one. This is the homopolysaccharides. Starch, glycogen, and cellulose all are made of glucose. All right, they are made of glucose. A starch. Next question is: a starch is a polymer of naturally glucose, according to this question given. This is one question. This one has been disturbed. Okay, leave it. Our next will be okay. Come to this question. This one, which of the following pair of nitrogenous base of nucleic acid is wrongly matched with the category mentioned against it? Can you read it now? Oh, so this is very big. There is a little problem here for the question. Okay, now you can read it. Which of the following pair of nitrogenous base of nucleic acid is wrongly matched with category mentioned against it? Guanine, adenine, purine, adenine, thymine, purine, thymine, uracil, pyrimidine, and uracil, cytosine, pyrimidine.
C.
See here,
Sir, number B.
wrongly match. Okay, wrongly matches. This is correct. Guanine, adenine, purine. This one is a wrongly match. This is purine. This is pyrimidine. Okay, so answer is this was given in CBSC 2008. Another question. Can you read it? Hello? In the DNA molecule, option A, the proportion of adenine in relation to thymine varies with the organism. There are option 2, there are two strands which run anti-parallel, one in the 5 5 prime to 3 prime direction and the other 3 prime to 5 prime direction. Next point is option, the total amount of purine nucleotides and pyrimidine nucleotide is not always equal. There are two strands, option 4, D. There are two strands which run parallel in 5-3 direction. Here in a DNA molecule, so here you have to select the correct one regarding the DNA molecules. Yes,
B.
A,
B.
B. Yes, this is, there are two strands. DNA molecules are, we know they are anti-parallel. Is it? They are anti-parallel and DNA have this, one end is 5 prime and one end is 3 prime. They have polarity in the polynucleotide chain, and when it is anti-parallel, they move in this direction, one opposite to. This is 5, 3, and this one will be 5, 3. So this one is correct. Say proportion of adenine in relation to thymine varies. It cannot, because adenine pair with thymine. So in a DNA molecule, number of adenine molecules will be exactly the same with the thymine molecule. Or if there is any, thymine is a little modified sometime, because in place of thymine, sometimes little modified unusual bases are also found. So adenine thymine will be always equal in number, it will not vary. And the total amount of purine nucleotide and pyrin nucleotide, there is also always the pairing is always between purine and pyrimidine. Purine and pyrimidine pairs. So naturally the total amount of the purine nucleotide and pyrimidine nucleotides also will be equal. So here written not always equal, it will be always equal. And this one, it is not parallel, so this cannot be the same. In the DNA molecule, you see this question, two questions are there. The form of DNA with 34 angstrom pitch with a rise per base pair of 3.4 angstrom is called. Which DNA it is?
B-DNA, sir.
B DNA. It is the normal DNA, that is this one is having 34 angstrom pitch. Pitch means that is one coil it moves. Ah, one complete circle. So that is called the pitch height. Pitch height is 34 angstrom, and there is a rise per base is 3.4 because at one pitch there are 10 base pairs. BP here is not blood pressure, this is the base pair. So the size of DNA molecule is written always in terms of the number of base pairs. What is the length of the DNA? This is, or the size of the DNA, this one, or it is sometime in picogram. Okay, in gram, that is in weight, or it is written in Dalton, that molecular weight total, as average molecular weight of a nucleotide is 300 Dalton. So 10 base pair in one pitch. So naturally the distance between the two base pair is 3.4, and this is the bDNA. There are different types of DNA are there. The common one, it is the right-handed coiling. The pyrimidine base which confirm additional stability to DNA over RNA is pyrimidine base which confirm additional stability to DNA over RNA is, in the pyrimidine, that thymine, uracil, cytosine. So in the RNA it is uracil, here it is thymine. So thymine is there in the DNA. This is another one, I think. In a DNA strand, nucleic acid bases pair with each other with the help of which bond? Bases. Bases pair with base pairing,
Hydrogen.
hydrogen bond. Okay. And in the nucleotide, in a polynucleotide chain, what is the bond? That one nucleotide is joined to the another nucleotide to join another nucleotide to form the polynucleotide chain. That bond is
Phosphodiester bond.
ester bond, or it is the phosphodiester bond. this bonds, etc. You have to remember. According to Watson and Crick model, DNA exists as a double helix. The two strands of polynucleotides are antiparallel to each other. The backbone is formed by sugar and nucleic bases, both A and B.
Option D, sir.
Option D. Yes, this is correct. It is double helical and they are antiparallel. This one here, sugar and nucleic acid does not form the backbone. Backbone is formed by sugar phosphate. Sugar phosphate. This is, say, if we say sugar, 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 there is phosphate, 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 phosphate. So this sugar is attached to phosphate, again phosphate attached to sugar, here with the phosphate, sugar, phosphate. So this forms the that side rod of a stair, okay, ladder, and the nitrogenous bases are at right angle to this axis and towards the inner side. So here if it is A, C, T, D, like this one form the backbone and this is the at right angle to this. Another one, if it will be there, then they will form. Okay, this side will be, say here the this side will be 5 prime, 5 end, because this phosphate it has with the fifth carbon of the sugar. All right, so fifth end, and this side it is the 3 end, and this one, this is 5 end, this side will be 3 prime end, and here A will be with T, C will be C,
G.
C will be with G, T will be with A, and here again C. And A and T paired with by the hydrogen bond, and there are 2 hydrogen bond here, CG, 3 hydrogen bond, 2 hydrogen bond, 3 hydrogen bond. This why say naturally the hydrogen bonds are weak bonds. Hydrogen bonds are, they are weak bond. Okay, but still the number of bond in the DNA molecules is very large. So they give a strength. And here, say in between A and T, there are 2 bonds. In between C and G, there are 3 bonds. So this portion is stronger than this one. This is weak and this is strong, because here it is they are joined by 3 hydrogen bond, it is by 2 hydrogen bond. And that's why those AT rich region, AT rich regions are weaker than G than A, sorry, GC rich region. All right. So AT rich region are, so there are in the DNA there is always there are some regions are AT rich, some regions are GC rich, and whenever during the DNA replication that the DNA strand, the two strand has to open. So opening usually takes place at the AT rich region. Okay. Oh, one question is still remain, but it's time. Okay, you can see some questions are there. This is of course NEET 16 one question. Few questions which are important. There is they were asked in the NEET. So this one question. This is AIPMT 2015, NEET 2016, and NEET 1 2016. So let's just say quickly this second question. Which of the following biomolecule is common to respiration mediated breakdown of fat, carbohydrate, and protein? This is pyruvic acid, acetyl coenzyme A, glucose 6-phosphate, fructose 1,6-biphosphate. This one is, I think we did it perhaps. This all the three things, the common compound in the Krebs cycle, that in respiration, this common compound where this fat, carbohydrate, and protein join, that one is the first one is acetyl coenzyme A. For the ATP system, it is a biomolecule, so this has been given from here. This question, which of the following biomolecule does not have phosphodiester bond? Nucleic acid in a nucleotide, amino acid in polypeptide, fatty acid in diglycerides, monosaccharides in polysaccharides. Which of the following biomolecule does not have a phosphodiester bond? Does have a phosphodiester bond. So this is nucleic acid in nucleotide. And the typical fat molecule is made up of fat. In the fat is fatty acid and glycerol. Okay. Okay, very simple fat is only glycerol. uh, and but the large fats, they have fatty acid. One is fatty acid plus glycerol. Okay, so this one glycerol and 3 fatty acid molecules. This was asked. So okay, today